Okay, we're going to start out with this. We're going to start out with a G protein. And it has GDP bound to it. I'm going to represent that by a circle to indicate one structural state. Now, this G protein is capable of ditching that GDP, releasing it, and replacing it with a GTP. So we'll say GDP for GTP exchange. So let's do that here. GDP goes out, and GTP comes in. Okay, that exchange process is ordinarily quite slow, but there are a set of regulatory proteins that can bind to the G protein and speed that process up 1,000-fold. What would ordinarily take 10 minutes takes a fraction of a second. And these proteins are called, we'll say, stimulated by proteins called guanine nucleotide exchange factors. Or GEFs for short, guanine nucleotide exchange factors. With a name like that, it tells you what these proteins do. The regulatory proteins that bind to a G protein and stimulate that GDP for GTP exchange. They speed it up. So in other words, these proteins tend to activate a G protein. Did you have a question back there? Do you have a question? Oh, okay. Sorry. Okay. So here we have another example of one protein binding to another protein and regulating. And in this case, the guanine nucleotide exchange factor regulates the G protein by speeding up that exchange and activation process. Okay, now once that exchange has happened, now we switch into this structural state that has the GTP on it. Normally this is active. And what it can do now is bind to and regulate various target proteins. So in this case, the active G protein is binding and regulating any of a number of target proteins. But all good things must come to an end, because G proteins hydrolyze their bound GTP. So we have Gtp hydrolysis. So the G protein is going to hydrolyze that GTP into GDP plus phosphate and expel the phosphate. And when it does that, it switches back to its original state. Now, this Gtp hydrolysis can be stimulated, once again, about 1,000-fold. Ordinarily, a G protein by itself takes about 10 minutes to hydrolyze that GTP. It's a really crappy enzyme in terms of how efficient and fast it is. But we can speed that up by binding another set of regulatory proteins. And these are called the Gtp Ace Activating Proteins. Or GAPs for short. And these GAPs bind to the G protein and stimulate that GTP hydrolysis. What they basically do when they bind is they warp the active site in such a way that the hydrolysis reaction occurs much, much faster than ordinarily would. So here is another example of one protein, the GAPs, binding to and regulating a second protein, the G proteins. So there we have it. Question? No? You did?
Just trying to figure out what that said.
Oh, what this says?
Further right.
Oh, stimulates GTP hydrolysis.
And also on the top.
Oh, on the top?
Right.
The exchange is stimulated by guanine nucleotide exchange factors.
Okay.
Those are a second set of regulatory proteins. So in effect, the GEFs, the guanine nucleotide exchange factors, what they do is tend to activate G proteins. The Gtp Ace activating proteins tend to shut them off because they cause Gtp hydrolysis to occur really quickly. So that's our monomeric G protein activation and shutoff cycle. Now when we look at the trimeric ones, we're going to see a few differences, but the basic pattern is similar. Once again, the alpha subunit of the trimeric G protein, that's the only subunit that can actually bind GTP or GDP. The other two, the beta and gamma subunits, don't. So let's take a look at the trimeric G protein cycle. I'm going to have to erase a few things here. Okay, so let's take a look at their activation cycle. I'm going to draw these G proteins as sort of the old style P symbol. Remember that one? Okay, most of you folks are too young for that. Okay. All right, so we're going to have a trimeric G protein. I'm going to represent it like this. Peace, brother. Okay. This one is the alpha subunit. And at this point, it has GDP bound to it. Here's the beta subunit, and here's the gamma subunit. Now, these are inactive in this state. Inactive. Okay, got that right. Now, once again, the alpha subunit is capable of GTP, of a GDP for GTP exchange. And that can be sped up because these guys have their own guanine nucleotide exchange factors, or GEFs. Okay, so what we're going to do now is GTP for GTP exchange. And we'll put GEFs, guanine nucleotide exchange factors, stimulate this. Okay, so that speeds the process up. So now we're going to have our P symbol here again. Here's our alpha subunit. GDP goes out. GTP comes in. Okay, so now we've done that exchange process. This is going to activate the trimeric G protein. And what happens when that occurs is this. The alpha subunit undergoes a radical structural change. And it changes enough that it pops off of the beta and gamma subunit, goes its own way. So now these things break up. You've got your alpha subunit with the GTP, and it's separate now from the beta and gamma subunits. And this alpha subunit now can regulate target proteins. Most of the time, the beta and gamma subunits sit there staring at you. There are some G proteins where the beta and gamma subunits can actually by themselves regulate other proteins. Normally that's not the case, but sometimes these guys can do regulation of different sets of proteins as well. But for the most part, they go off on their own way. Now we've got our alpha subunit, our free alpha subunit, GTP bound to it, it's doing its regulatory work. And here again, it hydrolyzes the GTP. Once again, this can be stimulated and sped up by the GAP, the Gtp Ace activating proteins, these regulatory proteins. So we'll put these guys here. So that can speed it up. So now we're getting our GTP hydrolysis, so let's do this. Here's our alpha subunit, GDP plus phosphate. And now we'll switch back completely. And release the phosphate. Okay, now once we've done that, the beta and gamma subunits can now reassemble with that alpha subunit. So everybody joins together. And here's the docking beta and gamma subunit. And everything joins together, and we're back where we started from. So the key difference here with the trimeric G proteins is that the beta and gamma subunits separate from the alpha subunit when GTP is found. The alpha subunit goes and does its regulatory function, and then when GTP is hydrolyzed, the beta and gamma subunits rejoin, and you're back where you started from. Those are the major differences here. So we see these activation cycles are quite similar to each other. You got a question back there?
What does it say on when you go down from Gtp hydrolysis? What does the box say?
Oh, this?
No, over here. That.
Oh, here? Oh, it says GDP plus phosphate. In other words, we've hydrolyzed the GTP, but we haven't dumped the phosphate out yet. So that's like partway through the hydrolysis process. Okay. Okay.